Well, good morning. It's great to be here with you, and thank you to BioLogos for having Scott and I come and present on this book. When I was invited to give this presentation, I was told I would have 25 minutes, and I thought, oh boy, 25 minutes to talk about population genetics to a non-specialist audience. So in the words of the famous philosopher Inigo Montoya from The Princess Bride, let me explain. No, there is too much. Let me sum up. So all that to say is what you're going to get here is just a flavor of what's in the book. But what I'd like to do today is just outline a few of the lines of evidence that are convincing to geneticists that our population size has never been below about 10,000 individuals. So this is sort of the scientific basis for the theological conversation. And I very much see my role as providing that scientific information such that it enables the theological conversation to take place. Okay. So April very ably and expertly in her plenary talked about some of the lines of evidence for evolution. This, of course, is Darwin's theory. That's been a very successful theory for the last 150 years. But it started as a hypothesis. And Darwin's observations led him to propose that species share common ancestors. So the basic idea here is that populations can become genetically isolated in some way, which means that genetic changes that are occurring in these now two separate populations through mutation or recombination of existing variation in those populations are not now being averaged across the two populations. So this means that differences can accumulate and average characteristics of those populations can change over time. So these differences may lead to new species. One analogy that I use in the book as a way to help non-specialists understand this process is an analogy to how human languages change over time. And how human languages change over time is actually a very good analogy for understanding how biological populations can change over time. So if you have two populations that speak a common ancestral language, those populations may become separated. Language changes. Now the slow progress of incremental change within those languages is now uncoupled between those two populations. So average characteristics can change. Differences might accumulate, and these differences may lead to new languages over time. So both species and languages, we can think of them as information systems that are copied in an imperfect manner. So there's not perfect transmission from one generation to the next of exactly that same information system, which allows for this incremental process of average characteristics changing over time. I talk about this in the book. I've talked about it in some blogs on the BioLogos website as well. If I put this up, even though we are Christians, and even though that is English of a sort, we have a hard time making out what we're seeing. This is actually John 1:29 from the West Saxon Gospels from about the year 990. There are letters there that we don't even use now. I love it, though, because it's got Middle Earth in it. Isn't that great? Okay, so if we look at this in a modern translation, it reads easily for us. So one might say, well, okay, how do we get from that to that? Well, of course, we know, anyone who's read the King James Bible or other translations would know that there's a whole host of intermediates, but this is actually a continuous population of speakers, and what we're looking at are discrete samples that have been pulled from this population at various points in time. And the analogy to biological change within a population is straightforward. Okay, so this shift in average characteristics over time of a language or a species means that separate populations might become mutually unintelligible to each other or become genetically incompatible if we're going to use the biological definition of a species. But the point is that the process is a gradient over time. So related languages and species can then be easily identified by biologists or by linguists by looking for shared features between the two that are in excess of what is necessary for that information system to function. This is an example I talk about in the book because part of my ancestry is West Frisian. These two sentences are pronounced essentially the same in both languages because West Frisian and English share a recent common ancestral population of speakers at about the year 400 on the European continent. Okay, so all that to say is we expect species to form as populations shift their average characteristics over time. Just like we expect languages to form, not because two individuals go off and start speaking a new language, but because populations, as they separate, have their average characteristics shift over time. So modern genomics very much shows us that our species was also part of that process of a population separating slowly from other populations. So not only did we arise through an evolutionary process, as April gave evidence for, but we also did so as a substantial population. At no time in our evolutionary history, it seems, has our population been below about 10,000 individuals. There's many different ways that you can assess this using genomics evidence, and the evidence from all these different independent ways of looking at that converges on this finding that we descend from a population of about 10,000. So I'm just going to briefly show you a few methods. Here's one that's reasonably intuitive. This is a large family group just representing individuals and their offspring. If we were to look at the present day generation, we might notice on a small segment of a chromosome different DNA variants that are present there. A's, C's, G's, and T's, that sort of thing. And we might notice that some of those variants are in a certain particular pattern. And then we might notice that that same little pattern is repeated in a number of individuals. Now, the most likely explanation for this pattern is that these individuals have inherited this little snippet of DNA on their chromosomes from a common ancestor that they share in the past. The other alternative hypothesis is that there have been independent recombination events or mutation events that have assembled that little snippet, but that's far less likely than inheriting it from a common ancestor, especially over short timescales. So what one can do is one can pick markers that are close together, and they stay together for a much longer time over many generations, or you can pick markers that are farther apart, which recombine more quickly. We know that these marker pairs are distributed among human ethnic groups in the pattern that we expect. So these different patterns of different variation correlate with what we know about human biology and our relatedness to one another. This sort of thing is what computational biologists love. Don't worry, there won't be like a test at the end or anything like that. But just to say, what we've done is we've looked at these sort of marker pairs in the human genome. In some cases, we've looked at huge numbers. So this particular study, for example, on chromosome 1, looked at about 1.3 million pairs of these different markers, and then simply asked the question, based on what we know about recombination rates and mutation frequencies, what population size in the past would be necessary to account for the amount of variation we see in this present-day people group. And in this particular case, you need about 2,800 individuals to be ancestral to this particular population group. And you can simply go around the planet and count up how many ancestors you need for the various people groups that we see on the planet, and you come up to about that 10,000 number. Okay, that one's fairly straightforward. I'm going to attempt to describe something that's a little bit more challenging to understand. I do spend some time on this in the book as well. And this really underscores the understanding that it's populations that separate. So when we draw on a phylogeny or a species tree, when we say two species are going their separate ways, they do so as a population, which means that they have variation within them as those populations go their separate ways. So what can happen is you can have that variation that's shared in a common ancestral population. That variation may not completely sort down to each descendant population. So here, if we're looking at humans, chimpanzees, and gorillas in the present day, humans and chimpanzees share a common ancestral population more recently than they do with gorillas. And in this particular example, we've seen some variants that go through to the human line, the yellow one to the human line, and the gorilla line, and the blue one sorting down to the chimpanzee. So in this particular case, humans and gorillas actually share, for this little snippet of their DNA, they're more closely related to one another than either is to chimpanzee. So this is something where they sit, where we have the sequences of individual DNA variants that sit at odds with the overall species pattern. Now that's pretty technical, but here's an analogy to help you sort of wrap your mind around it. And we have individuals from Canada. I'm from Canada. I'm in the States, and we have individuals from the United Kingdom here as well, so this is an apt analogy. Canadian and American English consistently group together as more closely related to one another. If you've traveled to Canada, you will have noticed that compared to traveling to the United Kingdom, that Canadians speak much more like Americans. even though it's not absolutely identical. One example, or just some examples here, different terms for different automotive vocabulary. You can see that Americans and Canadians consistently group together to the exclusion of British English. And that's because we share a more recent common ancestral population of speakers. Now, despite this overall pattern, there are certain features of Canadian English and American English and British English, that buck that overall trend. And one example is these OUR versus OR spellings. So in Canadian English, we retain the OUR spelling. That is also what the Brits do. So we have it right, and you guys have it wrong. I kid. Okay. So you might ask yourself, well, why is it the case that despite the overall main similarity between Canadian English and American English, why there's this particular feature that bucks that overall trend? And the reason for that is because these spellings were variable in the population in pre-colonization Britain and also in the immigrant populations that came to North America. You can actually look in some of the founding documents of the United States and see OUR spellings there. And the reason for that is that these spellings were still variable. There was still variation in this population when they were going their separate ways. And what we see is that the OUR spelling was retained in Canada and retained in the United Kingdom, and the OR spellings were lost there, whereas in America, the OR spellings were retained and the OUR spellings were lost. So this gives us some information. And this is something that's known as incomplete lineage sorting. The variability that was present in this population did not completely sort down to all of these descendant populations. So we actually expect this effect to happen for related species as well. It's actually something that we expect and we predict. So the presence of incomplete lineage sorting indicates that the source population had both variants, and that those variants were present in the ancestral population of the two most closely related populations. So this gives us information that those spellings were variable in pre-colonization Britain and also were variable in early immigrant populations to North America. If you have a way, don't worry, you won't be tested on the math. If you have a way to look back in time and say, this gives us information that there is variability in these populations, that gives scientists a handle to estimate the size of the population. And the reason for that is because the amount of variation that a population can hold is dependent on the number of individuals that are in that population. So this actually gives scientists a handle to look back in time and say, okay, we have an understanding of how much variability there was in these different populations. So based on other lines of evidence, we have estimates of these typical terms in this equation. The important term in this equation is n. That's an estimate of the population size of these different groups. We actually predicted that we would see about 25% of this effect between the human and gorilla genomes. So based on our estimates of this population size and this population size, we estimated that when we sequenced the gorilla genome, we would see this effect about 25% of the time. We actually observed 30%, which is in very good keeping with that prediction, and lets us know that our estimates of these population sizes were accurate. We've actually also done this with the orangutan genome now. The orangutan lineage would diverge prior to our divergence, prior to this common ancestral population. We predicted that we should have about 1.2% incomplete lineage sorting with that genome, and we actually observe 1%, which again is in very good keeping with our estimates. And you'll notice the population sizes that we're estimating here are quite large. They're on the order of 50,000 to 60,000 individuals. So as we go farther back in time, we see that our population sizes were actually larger, and that our shrinking to the 10,000 was likely in a human-specific sense. So it was not prior to our diversification from these other species. So these results show that humans have precisely the pattern of incomplete lineage that we expect if humans indeed share ancestral populations with these other species, and that the lineage leading to humans has maintained a large population for about the last 18 million years or more. So the population reduction, as I was just saying, to about 10,000 individuals seems to have occurred after we separated from the chimpanzee lineage. probably right around the time that humans, some of us, are leaving Africa and populating the rest of the globe. Okay. You've probably, however, heard that humans descend from somebody named Mitochondrial Eve. And some of you may have heard about Y chromosome Adam, her sort of male counterpart. And the analysis here shows that humans, all humans on the planet, do descend from one woman who lived in Africa, Mitochondrial Eve, about 180,000 years ago, and one man who lived also in Africa. I think current estimates are at about 350,000 years ago, plus or minus. And sometimes Christians hear this and they say, but wait a minute, didn't you just tell me that we come from a population of 10,000? So how is it that we come from we come from Mitochondrial Eve and Y chromosome Adam? Both of those things are true. And I'm going to briefly try to explain how that is the case. It has to do with how mitochondrial inheritance works. Mitochondria are the small organelles in our bodies that do energy conversion. And they have their own little circular chromosome that's inherited, solely passed down from mother to offspring, because sperm do not transmit mitochondria. Similarly, Y chromosomes, of course, only come down through the male line. So here we have mitochondrial inheritance going down from mother to offspring. Mitochondria that end up in a male are at a dead end because they can't be transmitted further. And Y chromosomes, of course, just go down through males. So if we look at a large group, again, we can now we can investigate what might happen to mitochondrial and Y chromosome variants in a population. So here we have 3 different mitochondrial variants in this large group, and that you can see that as we progress to the present day generations, that this one woman, her mitochondrial DNA comes to dominate this population, such that everybody in the current generations derive their mitochondrial DNA from her. Similarly, for the Y chromosome, we can see that all males in this generation inherit their Y chromosome from this man because of that particular pattern. Now, the question then about regular chromosomal DNA is inherited and passed on by both genders equally. It doesn't have this tendency to get lost because of a unique pattern of inheritance. So in this same group of individuals, we can now represent regular chromosomal variation with different colors. And we can see that that variation comes down to the present day from a large number of ancestors. So we all get our Y chromosomes from one man as a population. We all get our mitochondrial DNA from one woman as a population. And we get our regular chromosomal DNA variation from a group of about 10,000. So these things are both true. And it has to do with the different inheritance patterns of these different forms of DNA. Okay. So in summary, I've shown you that converging lines of evidence support the hypothesis that life on Earth is the product of an evolutionary creation, that God used the evolutionary process to bring biodiversity, including our own species, into being, and that for humans, it appears that he did so using a population, so that we have never numbered below about 10,000 individuals in our evolutionary history. So the question, of course, for the church is, well, where do we go from there? And that is why when I wanted to write this book, I didn't want to do it by myself, because it's very, very common for me to give a talk on genetics and have nothing but theological questions in the question and answer period. So I didn't want the book to be sort of the science and then me doing amateur hour theology. So I'm very pleased to have Scott on board, so you can ask him all the difficult questions, right? As I jokingly said to Scott when we were working on this project, I get to create all the problems, and then it's up to you to solve them all, right? So what I would argue is that a truly Christian approach to this topic must take Scripture and science seriously. These are both forms of God's revelation to us. Francis very ably discussed the 2 books metaphor in his plenary session. And I very much agree with that metaphor, that there cannot be conflict between what we see in science and what we see in Scripture. But these are both science and exegesis are human activities, so they're not going to be perfect, right? So we need a humble approach. We must recognize that both human interpretation of Scripture and science are fallible activities. So if there's a conflict, it's a perceived conflict, because there's no inherent conflict between God's two forms of revelation. In the case, however, where we have scientific evidence that remains strong, despite decades, or in this case, centuries of examination on the evolution question, Christians have always had warrant to reinvestigate their interpretations of Scripture, because general revelation is also a form of God's revelation. So, and for my part, I see that the role of believing scientists in this conversation is to present the church with the best scientific information available, such that the theological conversation can be informed by what we're seeing in this form of God's revelation. Thank you very much.